Darwin reigns supreme. The theory of evolution is an absolute paradigm that holds the entire intelligentsia captive. There are many individuals who are unwittingly prisoners of this deception, but these individuals will soon be free because now Darwin's powerful illusion can be rigorously and scientifically falsified.
What we've just heard are not the words of a pseudoscientist, but those of Professor John Sanford, one of the most prominent geneticists in the world. But how did John Sanford, a scientist of global renown, come to formulate a theory that refutes decades of study and experimentation by his peers? To fully understand the revolutionary scope of this theory, we need to take a step back and start from the beginning of this story. Today, we'll discuss the theory of genetic entropy, a complex and fascinating theory, but also an extremely divisive one. As one can easily imagine, Professor Sanford's theory has sparked very harsh reactions in the scientific community. Denying the validity of neo-Darwinian theories has cost him fierce criticism and the distancing of a part of the mainstream scientific world. But what did the professor discover that was so significant it led him to reject years of research and study? John Sanford graduated in 1976 from the University of Minnesota, where he then specialized in plant genetics. From 1986 to 1998, he was an associate professor of horticultural science and was also an honorary associate professor of botany at Duke University. Over the years, the professor has published over 70 scientific papers and has registered an impressive 32 patents. At Cornell University, Sanford, along with his team, developed the biolistic particle delivery system, the so-called gene gun. The gene gun is a method for introducing foreign DNA into a cell with the goal of creating transgenic organisms. This is currently the most used direct transfer method for transforming plant cells. The professor has been awarded the prestigious Distinguished Inventor Award twice for the significance of his research. In addition to this, the professor has founded 2 biotechnology companies, Sanford Scientific and Biolistics. Well, having made this quick introduction that certifies the professor's expertise, let's dive into the heart of the discussion. And since Sanford is a geneticist, we must begin by talking about the genome. The genome is the complete set of genetic material of an organism, contained in the nucleus of its cells and organized into structures called chromosomes. In the case of humans, the genome consists of about 3 billion pairs of DNA bases, which contain the information necessary for the building and functioning of the body. This information is encoded in the form of genes, which are specific segments of DNA responsible for producing proteins and other functional molecules.
The genome can be likened to an extensive collection of encyclopedias that serves as a detailed manual for the construction of both a single human cell and the entire human body. Although it may seem similar to a computer program or an operating system, the genome is actually far more sophisticated and advanced than any technology developed by humans. Each year, we discover new wonders within it, realizing that it is even more astonishing than we had previously imagined.
The Human Genome Project was one of the most ambitious scientific endeavors in history. Officially launched in 1990 and completed in 2003, the project aimed to map all the genes of human beings and determine the sequence of chemical bases that make up human DNA. This project involved numerous scientists and laboratories around the world and represented a turning point in genetics and molecular biology. The genome is at the foundation of everything we know and call life, but according to Professor Sanford, the genome adheres to a universal law, a law with which we all grapple daily: entropy.
Entropy is a concept that is interpreted and defined in various ways depending on the discipline. While physicists may have a technical definition based on thermodynamics, engineers or information scientists might see it from a different perspective. But at its core, entropy represents a simple universal truth: the intrinsic tendency of everything to disperse, deteriorate, decay, and approach an end. It's not an abstract concept or far from our daily experience. It's something we observe and live through every day. Many recognize it as Murphy's Law, the idea that if something can go wrong, it will. Whether it's a new car that begins to show signs of wear over time, or a house that requires ongoing maintenance, entropy is that constant and pervasive force against which we struggle daily in our lives.
If everything tends toward chaos, from our homes to the outside world, to the entire universe, does this universal law of disorder also apply to living beings, and specifically to our genome? Let's try to simplify a concept, that of aging. In your body or mind, we have about 3 new mutations every time a cell divides, which at the moment, according to scientists, seems to be one of the reasons we age. The reason all our systems begin to break down is this mutational process and the accumulation of degenerative mutations in our genome. This is why there is a maximum limit to lifespan. But according to Professor Sanford, the problem is not confined to the individual, but is even larger.
Decades ago, a renowned geneticist predicted that with just 1 mutation per person per generation, the degeneration and extinction of the human species would be certain. Recent discoveries show that the mutation rate is much higher, with over 100 mutations per individual per generation. This implies that every individual carries about 100 mutations not present in their parents, and in turn, passes them on to their children. With each generation, about 100 new mutations are added to the human genome, and this is a conservative estimate. It's believed that the mutation rate could be even higher. In the current landscape, each of us carries tens of thousands of negative mutations. The reality is that we are all mutants, and significantly so. Statistics show that 2–3% of newborns have obvious congenital defects. This percentage represents just the tip of the iceberg. Most mutations are subtle and do not produce immediately visible effects, just like the rust that slowly forms on a car. We don't see every single atom oxidizing, but it's the cumulative effect of these atoms that produces the visible rust. In terms of the accumulation of genetic load, we have identified over 6,000 Mendelian genetic diseases in humans. A Mendelian genetic disease is caused by a mutation in a single gene, like hemophilia, which produces an obvious effect and can be traced through generations.
So, according to the professor, every person passes on a series of mutations to their children, generation after generation, and these mutations, while minute and not immediately apparent, accumulate over time and may ultimately jeopardize the functioning of the entire DNA. However, neo-Darwinists argue that natural selection would intervene to halt this drift by eliminating negative mutations and allowing room for positive ones.
Personally, my specialization is in genetics, with a specific focus on plant genetics, selection, and enhancement. Having worked in this field for many years, I feel qualified to speak authoritatively about natural selection. Natural selection undoubtedly plays a role, but its scope of action is extremely limited. Although it's possible to select out a particularly harmful mutation, it's difficult to identify and select the typical deleterious mutation, as its effects are often too subtle to be detected. It's like the atoms of rust on a car. They're so small that we don't notice them, and consequently, we can't eliminate them. On the other hand, it's possible to select a particularly advantageous mutation, for example, one that helps an organism adapt to specific environmental conditions, like resistance to an antibiotic. However, what's virtually impossible is selecting a single nucleotide in the genome, which represents only a minuscule fraction of the overall genetic information. The sum of all these small mutations that are passed down over time, if you closely examine the specialist literature related to population genetics in recent years, you'll find that many eminent geneticists recognize inconsistencies in the neo-Darwinian theory. These criticisms, while known within narrow academic circles, have not been widely disseminated to the public or the broader scientific community, remaining almost like a trade secret. The main logical flaw in the neo-Darwinian theory lies in the relationship between mutations and natural selection. If the mutation rate is high and most of these mutations are harmful, there is no selection mechanism that can effectively offset the influx of negative mutations. Natural selection can only eliminate a fraction of the most harmful mutations, but it can't counter a high rate of negative mutations. A useful analogy to understand this concept is that of a boat taking on water. If water enters the boat faster than we can bail it out, it will eventually sink. Similarly, natural selection can only handle a small percentage of harmful mutations, but it can't compete with an excessively high mutation rate. In addition to this, it has been found that beneficial and harmful mutations often lie close to each other on a chromosomal sequence. This physical proximity makes it difficult for natural selection to distinguish and select between positive and negative mutations.
The professor's reasoning, if we trace back in time, inevitably leads us to a distant era when humans, and not only humans, made their entrance into the world with an intact genome.
A renowned evolutionary geneticist has pointed out that, genetically, we are inferior to cavemen. Despite being a fervent evolutionist, he acknowledged human genetic entropy. But this isn't an absolute novelty. If we examine the cultural traditions of various societies, many suggest the idea of genetically superior ancestors. Ancient scriptures, for example, offer an interesting perspective on this topic. Analyzing the Old Testament, we can trace the longevity of the patriarchs. Starting from Noah and passing through the generations up to Jesus Christ, we can observe a pattern. This pattern illustrates a decrease in lifespan, forming what we might define in biology as a curve of biological decay. When I first read these parts of the Bible as a young person, I thought the numbers were exaggerated. But observing the consistency of the data, one can notice a precise structure. If someone wanted to invent such numbers, would they really create such an accurate decay curve? This pattern suggests that the scriptures record a real biological degeneration over time. Our genome is undergoing alterations, with its sequences being modified due to mutations. These changes affect the functioning of our bodies. Unfortunately, we are all susceptible to these changes, which affect us both as individuals and as a species as a whole.
With DNA sequencing, large regions have been identified that do not code for proteins and seem to lack a specific function. These regions, called junk DNA, are considered by some scientists to be evidence of the evolutionary history of our genome and refute the idea of a genome that appeared whole and functional.
When the genome was initially mapped, much of it was believed to be junk DNA, a dominant concept around 2007. However, with the completion of the second phase of the genome project, known as the ENCODE project, light has been shed on many previously unknown functions of the genome. The discoveries revealed that over 90% of the genome is actively transcribed, suggesting a surprisingly high level of functionality. This indicates that we need to study even more closely to understand the functions of the remaining parts of the genome. But even more astonishing was the discovery that the genome contains multiple levels of overlapping messages. It's like reading a book where each chapter has hidden meanings embedded beyond the obvious meaning of the words. Incredibly, it was found that more than 50% of the genome is read on both DNA strands, not just one as previously thought. This is comparable to reading a sentence forward and finding one meaning, then reading it backward and discovering another message. Essentially, it's a form of data compression at a level of sophistication never seen before. This raises doubts about the possibility that such complexity could emerge through random changes. Even in the world of computer programming, every bit of code, whether a 1 or a 0, is there for a reason. Nothing is left to chance. Similarly, the concept of junk DNA is increasingly being questioned in light of new discoveries.
If we accept Sanford's theory and refute the idea that mutations and natural selection are the forces that have shaped and refined the genome, we are faced with an inevitable dilemma. Was the genome created by an external entity, or did it emerge spontaneously as a result of mutations and evolution? Professor Sanford seems to have no doubts.
It's becoming increasingly challenging to explain how the genome could have formed, especially considering its multiple levels of overlapping information. Our best computer programmers struggled to conceive codes of such complexity, making human technology seem rather rudimentary in comparison to what we observe in the genome.
Sanford's research and his scientific stature make the theory of genetic entropy both fascinating and full of questions. But it's also true that it refutes everything we thought we knew about evolutionary processes and opens up scenarios that are somewhat unprecedented. Is humanity, as well as all life on our planet, the result of intelligent design or simply a product of chance? For now, we'll stop here, but let us know in the comments what you think, and if you enjoyed the video, subscribe to the channel. Until the next story.